explore my research credentials on bio-nanomaterials, that is especially in biopolymers, bioceramics, carbon-based materials and metal oxides for healthcare applications, in particular chronic wound healing applications. So the outline of the presentation covers the primary introduction to the chronic wounds, overview, significance and properties of the wound healing materials. Also there are 4 different types of materials which contributes to the more positive wound healing, and their theoretical and physiochemical and biological applications will be discussed. So initially the wound healing is a major and impactful area in the biomedical engineering because it has a many role in the lifespan of the chronic or diabetic patients. The diabetic or chronic wounds have a slower and low level of healing phase because the growth factors and the protein regenerations are low in the chronic wounds. Due to low growth factors, reduced androgenous formation and also tissue necrosis, that is tissue damage, is also possible especially in the diabetic wounds. So with these complications, we will be looking forward to a better biomaterial, that is a bio-nanomaterial with having a unique properties like non-toxicity, non-allergenic to the skin or any surface of the patients, and also antibacterial, antimicrobial, anti-inflammatory and antioxidant properties, and also with the collagen deposition. So some of the biomaterials like bioceramics, biopolymers can be used for these kind of applications. So I am here to discuss about 4 different types. First one is a bioglass. It is a matrix of silica combining with calcium phosphate and sodium. So this bioglass is also have 4 different types like 45S5, 58 bioglass, 68S and 78S. The number mentioning is 45% of silica and 58% and 68% of silica. So first 45% is silica and next calcium and phosphate is 24.5% and others is phosphate is 6%. So with this I am going to combine one biopolymer, that is chitosan, and also a graphene oxide material and a metal oxide and carbon quantum dots, which enhance the wound healing in a faster manner and also without any allergic forms. So the first phase of work is bioglass combined with graphene oxide. As the bioglass has many properties like cell proliferation and androgenous formation, it lacks in antibacterial activity. So induce the antibacterial activity, I have made a composite with graphene oxide. It is a good antibacterial material, mechanical stability, and also the functional materials of graphene oxide are covalently linked with the protein and other materials in the blood and other health infections. So first I have synthesized bioglass by Sol-Gel method and graphene oxide by a improved Hammer's method, and both these materials are composite into a wet chemical reduction by hydrogen hydrate. After the physiochemical studies, the structural studies like XRD were shown with the graphene oxide at 001 and 002 plane, and BG sodium. Here I have to mention that bioglass with sodium and without sodium was synthesized, because as I said earlier, the bioglass contains silica, calcium, phosphorus and sodium. The role of sodium in the bioglass may or may not have a negative impact on wound healing that will lyse the blood. It will rupture the blood cells. So I have intended to work on both sodium free and sodium with sodium bioglass. So here the sodium bioglass with sodium and without sodium as shown as silicon is calcium silicate phase and sodium silicate phase with silicocornate and combined phase. Further, the SEM studies shows as the bioglass has different materials, metal ions, it has shows more different morphologies like spherical, flakes-like and also a sheet-like rod type. So, and the graphene has shown the stacked sheet-like structure. With the combination of these, the bioglass and graphene will be a formation of a floral and flake-like structure, which can be and good for antibacterial activity. So, and the antibacterial activity were studies for all these 4 samples with Streptococcus and Pseudomonas bacteria has shown the bioglass without sodium and the bioglass without sodium graphene oxide composite has shows a good antibacterial activity. In the same way, the hemoclot, because the wound healing has 4 major phases. First is hemoclot, the blood clotting. Then is the cell proliferation or cell multiplication, and in inflammatory phase and the remodeling phase, that is re-epithelialization phase. In that, the clotting phase is more important. Here the BG sodium free composite shows 3 minutes of clotting time than other composite materials, and also I have combined it with the lysis. So this is the lysis results. Here also the sodium BG, sodium free BG has shown less lysis than with sodium BG. And the DPP HSA, because we have to determine the antioxidant property as the materials have more hydroxylic and carboxylic group. The DPP HSA was assisted and shows a better inhibitory activity against the radicals. Then the in vitro studies like cell viability is up to 98% and the cell proliferation is conducted for 72 hours. And the apoptosis assay shows BG sodium free and GO up to 24 hours with the more cell attraction with a bright green fluorescence with the wound contraction. Here we have gone for in vitro scratch assay, but it's at up to 24 hours we checked the wound was not completely closed. So this is because of the time duration we have limited up to 24 hours. So due to some technical reasons, so this is how the this phase is concluded that incorporating graphene oxide with bioglass for antibacterial and mild wound closure property. So with these drawbacks, I have gone for the next work, that is the work has been published in scientific reports. The next phase of work is I have combined bioglass with a chitosan biopolymer. There are some 3 or 4 polymers like chitosan, collagen and alginate, which are very useful for soft tissue regeneration. In that, we choose chitosan for cost effectiveness and easily synthesis process. So here also the same concept, bioglass with sodium and without sodium was introduced. The synthesis process, chemically cross-linking method used for chitosan process, bioglass solvent method, and both the composites were prepared by wet chemical method, and these types of scaffolds were prepared for lyophilization technique at 800 degrees Celsius. So the characterization studies, XRD, the chitosan shows the amorphous nature, and the combining with BG, the sodium and calcium phosphate silicate was embedded higher. Further, with the FTIR pattern, the amide 1 and amide 2 NH bonding chitosan were majorly attached to the bioglass of silica and calcium. And here the FSM result shows chitosan as a sheet with the BG incorporation. That is some, it's like a layer, it was deposited on the chitosan sheets. And in the contact angle, the chitosan has shown hydrophilic and others have shown hydrophobic properties. In the surface area, the chitosan is a semi-mesoporous material, chitosan alone. But when combining with the chitosan and BG, it has shows a type 4 isothermal mesoporous material which can able to interact with any blood bioactivity to combine out for the porous type of material. And this is the inflammatory analysis, because any type of material need to be avoided with antibacterial in the first case. Next is the inflammation or swelling property. To suppress that, many kind of proteins and enzymes to be involved. So the COX, LOX, MPO and nitric acid were studied here. These are all the enzyme inhibitors. So enzyme inhibitors or material act to prominently act in that, these are all controls. So material has prominently act to the level of the surface inhibitors. So here The schematic represents the NOx and COX with high indexes and nitric oxide level as low, because the nitric oxide will cause the inflammation to be produced in the tissue or nucleus level. So next is the in vitro studies. Here we gone for the confocal images of the scaffold. We can see here the 2D profile with the in-depth cell penetrating into the scaffold, and this is a wound contraction. Here the in vitro scratch assay shows complete wound closure at the 18th hour with the chitosan and BG sodium free sample. As the chitosan shows longer hour for closure, the composite has shown 18 hours. So this is the schematically admitted. So after these in vitro studies, we gone for an in vivo application. That is how the scaffolds act for animal studies. Here we have taken only two set of samples, pure chitosan and chitosan bioglass without sodium. So first, in the rat model, we have, so this is a diabetic wound model. So in the rat model, we have induced diabetes and we have treated that rat for 4 weeks. After that, we have made a wound on the first 0th day and we have placed the scaffold. At the initial stage, there is a blood clotting as occurs initially, and from the bioglass, the release of ions was noted in the upcoming days. The blood clotting was done. After blood clotting, angiogenesis and re-epithelialization was done. After that, the inflammation or swelling phase is also depressed here. Further, a new cell formation was formed at the 7th day itself. So it's a positive sign for a scaffold that within the 7 days, a wound has been healed by a scaffold. So the part of in vitro has been published in RSC Biomaterial Science. So these studies are already discussed by Professor Nebu sir. We have already collaborated with him for the animal studies. Here our product has shown best skin sensitization activity, and also there is no allergic or any other external factors like weight loss or skin pigmentation or skin decolorization or any other toxic substance has evolved in the surface. Here, for the diabetic wound animal model, I have shown the HNE staining and Masson trichome staining. Here, the re-epithelialization cells were formed at the higher concentration for chitosan BG sodium free, and the collagen formation deposition, that is for skin regeneration, has been rapidly occurred in the higher concentration for same chitosan BG sodium free at the 7th day itself. So this is the reason for the wound closure on the 7th day. So the work has been published in Biological Macromolecules elsewhere. So beyond this, we have concluded that I have a scaffold that can heal a wound in a particular phase. So but this is not enough. What happens in the wound healing phases? How the clotting, any infection has occurred in the phases of wound healing, at what the time proliferation is occurs, and what the time skin remodeling is occurs, and what happens when the scar is occurs. So I need to monitor my wound healing phases, that is tracking of a wound healing. For that I need a fluorescence material, and I need to image how to control or how to assess the wound healing phase, tracking of the wound healing phases. So I go for a carbon based materials which have a more photo stability and photo activity. These carbon quantum dots are not chemically synthesized. It's all derived from a biomass based with extraction method. This is the two types of groundnut shell and pistachio. The waste products has been extracted and the carbon quantum dots was prepared by a extraction and hydrothermal method. So the XRD pattern for both pistachio and groundnut shell CQD amorphous nature, and both has the Raman spectra of more in D band, because the defect band is more due to the silica content in the groundnut and pistachio. So the FTR spectrum also shows the carbons, hydroxylic and carbonylic group were interacted with nitrogen and oxygen and hydrogen. Further, This is the synthesis process. This is how the ground nut shell and pistachio were extracted from after cleaning, and we have hydrothermal method with the carbon quantum dots. So with the excitation of UV excitation of 309 and 370 nanometer, a contour mapping was drawn with 440 and 56 nanometer. Here the pistachio shell shows blue color and the groundnut shows green color fluorescence under the UV lamp. So the temp images shows a fine carbon quantum dot structure, and also a discrete level of energy levels was supposed to be there in the carbon quantum dot. That has been verified. Further, the same in vitro studies has been replicated for the CQDs. The cell viability of probability 98%, hemolysis is only 3% and 5%, and protein denaturation has occurred, and also the gene expressions for IGF and collagen. Insulin growth factor and collagen genes have showed a stimulatory effect for the CQDs. So, and also with the zebrafish larva, we have imaged how the CQDs shows the growth development in the zebrafish. Here, the eye region and the oak scale region has shown a bright blue fluorescence than other region with the help of the CQDs at 530 nanometer. So this is how the CQD has worked for the bioimaging application. So the work has been published in photochemistry and photobiology as well. Next, only CQD can only track or bioimage my wounds or any surface of wounds. But I need, with the tracking, in this simultaneously tracking, I need to heal my wound without any adverse effects. For that, again I gone to my back material, that is bioglass, chitosan and gold nanoparticles. So here, why gold nanoparticle? It's a antibacterial activity, biocompactable and high photostability. So with these three properties, it's a combination of binary and ternary materials. One is bioglass and gold nanoparticle, the other is bioglass, gold nanoparticle and chitosan composite. So first, initially we have tested for antibacterial and drug loading ability. So as usual, because of gold and chitosan, the antibacterial activities is achieved as soon. As in the drug release profile, we have introduced metadienosyl drug. It is an antibacterial drug. With this, 4 kinds of kinetics has been studied: zero order kinetics, core well kinetics, and also HE model kinetics. In that, the zero model kinetics has shown correlation efficiency of 0.9 R square, which is well suited for the bioglass gold chitosan composites. So, but other methods are not suited because due to the dissolution factor of bioglass and gold nanoparticles, and also the non-diffusive nature of chitosan. So the materials stick with the zero order kinetics. So the zero order kinetic has shown an enriched drug delivery property with more longer and stable drug delivery in the site. So this is how the work has been. I have concluded my summary as I have working on the wound tracking application. I need to build my material for the enhanced phase of wound tracking with bioimaging like fluorescence or confocal imaging. So here I acknowledge my supervisors, my mentors for the constant support on my work in the research outcomes, and my collaborators Dr. Sabu sir, Dr. Nebu sir, and also Sendil Sundaram for the scientific report papers. And these are all the different funding agencies I have worked on for characterization and other analysis. So these are the outcomes of my current research work. As I am a EEE holder in the UG, I am also working on biosensors and energy and environmental applications like supercapacitors, etc. Thank you. Thank you for the opportunity.
Long live India and France friendship.